Our OICR Mondays discussion topic today is entitled An Open Science Approach to Target DIPG, an incurable pediatric brain cancer. DIPG stands for diffuse intrinsic pontine glioma, and it's an aggressive tumor of the brainstem in young patients. The format for today's session will be an introduction from the moderator, followed by 3 short presentations from each speaker. Each speaker represents an OICR research theme. After the presentations, there'll be a discussion session at the end where you can join, where the audience can participate. We're using Zoom webinar, so please raise your hand to ask a question and type it in the Q&A box where it'll be answered by one of the panelists. Now I would like to introduce today's moderator, Dr. Aled Edwards, who will guide us through the event today. Dr. Edwards founded and leads the Structural Genomics Consortium, which is a public-private partnership in the biomedical research and pharmaceutical sector. The SGC generates research tools and knowledge to support basic science and early-stage drug discovery, and since 2004 has placed all its research output into the public domain without restriction, including without patents as a core principle. Dr. Edwards is also a professor and Temerity Nexus Chair of Health Innovation and Technology at the University of Toronto. Aled, thank you for joining us, and I'll turn it over to you now.
Yeah, thanks very much, and it's nice to be here. And we have an interesting afternoon ahead of us with a clinical expert and then 2 trainees that'll talk about the genetics of DIPG and the efforts into drug discovery on DIPG. And so, in general, the problem we're trying to tackle is DIPG is just representative of a number of cancers that are so rare that they can't attract commercial interest in drug discovery. So they're so-called areas of market failure. And so the choices that society has are to either ignore them, which is basically what has happened by default, or to determine or figure out an alternative path to try to invent medicines for the patients, and in this instance, it's children. And so DIPG, I'm going to sort of frame, we'll go deeply into DIPG today, but remember, it's a model for all cancers that have very few patients and that have a molecular mechanism. So DIPG, we selected as the model to explore alternative business models, is because it has very clear genetic underpinnings, and that was only discovered in the last decade or so when better sampling methods that came to be. And among the various genetic mutations that I'm sure we'll hear about today in more detail in the talks was one of the subtypes has an activating or a gain of function mutation in a protein kinase. And protein kinases are a precedented drug class. It's, in quotation marks, a simple drug discovery project in that we know the target is druggable. There's lots of precedent, and the mechanism to create a kinase drug is precedented in the community. So we thought, well, here's a great example of a molecular mechanism, a druggable target, and a clear therapeutic strategy. I say therapeutic strategy, but I don't really mean that. What I really mean is a mechanism testing strategy. For although activating mutations of ALK2 are found in the tumors, it doesn't necessarily mean that the kinase is required for the tumor growth once the tumor has come to be. So if we were to invent a molecule that could be drug-like for this kinase, the real question is, does inhibiting the kinase in a tumor, in a person, have a therapeutic benefit? And that's a hypothesis. It's almost a basic research question, okay? So to answer that question, you need a very good molecule, a potent molecule, a selective molecule, a safe molecule, a molecule that engages, that binds to the target in the brain, in this instance, in the brain of the children, and that is drug discovery. I mean, the molecule that we would have to make to test the hypothesis has to conform to our definition of what a drug would be. But I emphasize the point in doing this experiment to try and invent this kinase inhibitor is to test the hypothesis. And if you frame it that way, then you think, wow, this is really a research question. It's not a drug discovery question. And I think that framing is super important in developing the business model to invent this medicine, because when you're talking to potential funders, albeit if you talk to people who are very interested in the disease, it'll be framed as a drug discovery problem. In reality, it's a basic research problem that the community can get behind. And so if we're going to frame it as a basic research problem, and there's no big magic pot of money commercially to make it happen, the only mechanism to actually get this drug or this molecule to a place where it's sufficiently advanced that it's safe enough to go into people is to, in quotation marks, crowdsource the science, to get contributions from scientists who are pharmacologists, who are chemists, who are biochemists. And not all these people are in one place. So to be able to do that, you need to have some collaborative mechanism where you can get input from multiple sources. And if you ever have to deal with any collaborations like I have, even getting one institution to agree to your legal terms, you might as well wait for Halley's Comet to come again, let alone multiple of them. And so really the only tactic, the strategic business decision to enable this multi-institutional collaboration, the default, it has to go to open science. You have to take potential monetary gain completely off the table, or else you're never going to get these multi-institutional collaborations to work. So open science is the default, what we ended up with, but that wasn't the objective. That was the business tactic, because one would never have been able to aggregate these folks together in one room. Now, Michael Taylor said something funny, and I really think it's true. It's like, professors and clinicians are like kindergarten children. If Johnny gets more candy than oh, am I upset? But if you get the same amount, even if it's zero, we're happy. And so that was in fact, the mechanism that we entered into this, is that there would be no financial gain by anybody. So the company that we created to hold the intellectual property, which happens not to be a patent, but we can talk about that later, is owned by a charity. And the charity is 100% owner of the thing, of the company, and the charity purpose is to give money away or not make money at all. So we can make it affordable if we ever do. If this hypothesis testing molecule happens to work, then we can, because we have no investors looking to maximize the profit, we can make it affordable. And so when we embarked on this open science experiment, seeing if it would enable folks to aggregate, it was generously supported by OICR at the beginning and led by the OICR team, but it was augmented with about 6 different companies. Charles River is a major contributor. They are, in fact, assuming responsibility for one of the whole chemical series that's undergoing here. We have fantastic collaborations with CAMH. We have fantastic collaborations with the University of Toronto, the University of Oxford, with Penn, with a number, probably about 6 or 7 universities and 6 or 7 companies, all helping advance the idea without any expectations of financial return. And again, that was critical, or else it'd be 0 collaborators. And so we have 2 chemical series that are being advanced. So you want a drug-like series, and just in case this one doesn't work, we have another one. They're at the lead stage, which means that they're pharmacologically okay. We could probably, we're trying now to choose the best ones that get into the brain and engage the target. We're brought in a team from North Carolina now to help us manage the project as well. And I think that the objective of this is to be able to get to a point where we have a molecule that we think is the one that we would like to do the further pharmacology on. Because in the drug discovery cascade, what we're doing is the inexpensive part, although it's been expensive. The next step is rather formulaic. You know, you have to make kilos of the compound that you choose and do toxicology on that one. The clinical trial part, I think, will be facile because there are a lot of engaged parents and patients. So that'll happen, but we need to make a safe molecule that we know gets into the brain and we know inhibits the kinase in the brain. And that's the challenge now. The project went superbly well until about 3 years ago. Then COVID hit, and then it went on a hiatus. And now it's coming back, and we started again to do some experiments on some of the compounds that are starting to be produced. And I think Hector will tell us about one of the series. So in sum, the DIPG project that we undertook in the open was a project that is aiming to make a molecule that's safe, effective, selective, and gets into the brain and engages the target as a public good. We have no patents on it. The idea that we'll be able to share the compound with everybody. The intellectual property is, I can explain later if anyone's interested, is the regulatory data. The molecule are being worked on by a number of chemists around the world. There's a series being worked on in England by the Charles River folks. We continue to get input from industry that helping us run assays and doing some chemistry. And presumably in the next year or two, we'll hopefully have identified the clinical candidate that will be ready for going into IND-enabling studies. And so I think it's a super interesting experiment in the business because, like I say, if we don't invent a new business model for the many, many, many cancers that have so few patients, they'll be ignored by industry. And so the whole, oh, why don't you file a patent and raise some venture capital money and something, it just won't work, because if there's no market, there's no rationale to invest. This has to be undertaken by the public sector. And if the public sector is going to undertake it, we should charge affordable pricing at the end of the day. And that's the model. Anyway, there's lots of time for discussion later. And I zoomed over the DIPG and the genetics and the drug discovery, because that's what our speakers will be talking about. So right now, we'll shift to the speakers. And it's a pleasure to introduce Dr. Das from SickKids. And he is a clinician scientist who's, among his many things that he does, is he looks after and cares for some of the pediatric patients that have brain tumors, including this one. And he'll give us a sort of précis of what's going on in the world of DIPG from the clinician's point of view. So thanks a lot. Over to you, young man, and I'll go on mute and stop my video.
Thank you, Aled, for that exhaustive introduction, which was very comprehensive and allows a set stage for me to see how we as clinicians and as clinical trialists are trying to navigate this difficult path, which was alluded to. And thanks to OICR and Cynthia for allowing me to present the clinical perspectives. So I'm a neuro-oncologist, and I work with children, unfortunately, with this deadly disease that usually presents on the MRI like this. You can see the whole of the pons is sort of involved, and you can see the white thing here. And this is something that we need to sort of tackle and discuss how we tackle this best. So as was mentioned, DIPG is diffuse intrinsic pontine glioma. But just to get our nomenclatures correct, it is now an expanded spectrum of tumors that not only includes tumors in the pons, but tumors with the same biology in the other midline structures that you can see over here. So it's now called diffuse midline glioma, or DMG, and it comprises 10 to 15% of childhood brain tumors. And as was mentioned, it's a relatively rare tumor because we have only 300 odd patients in the US and 30 to 50 per year in Canada. But these are like really young patients who usually present between 6 to 7 years of age. The pons, as you can see, is the most common site, but there are other sites, as I mentioned. And I want you to remember that there are some patients who are on the older age of the spectrum, as you can see from here, maybe in their teens and maybe in early adulthood, who have a preponderance to develop this tumor in other locations like thalamus. And it will be important when I talk about clinical trials, success versus not success, of why that plays out and why that is important to remember. It is an aggressive cancer, presents very rapidly with a symptom of only 2 months, and it's a devastating diagnosis for parents as well as us physicians who see them presenting to us. So I know there will be talks about biology, but just to mention that it is defined by a specific molecular pathway or a molecular alteration, which is the H3K27 alteration that is present in these diffuse gliomas. The mutations are listed over here. It is important, and Cynthia will always tell us that it's not that the pathology can be very variable, but it's the molecular driver that determines the biology and the outcome of our patients. There are other subsets that have a similar mechanism, but not exactly the same. They are listed over here, and they can have EGFR mutation rarely as well, especially the thalamic tumors. And there are a set of cooperating mutations that people will definitely talk about today, and some other mutations which are rare in this subset of tumors that really distinguishes them from other high-grade gliomas or glioblastomas that we see in children and young adults. So moving on, we as clinicians know that it's a devastating tumor because of the location that I showed you. It's in the pons, it's diffuse, it's in the midline, so it can't be taken out. Chemotherapy, the traditional one, temozolomide, which is used in all high-grade gliomas, was shown in historical trials to be really ineffective. Radiation is the only thing that works, but that results in a median survival of only 9 to 12 months. And at the end of 2 years, there are only less than 10% of patients alive, as you can see in the survival curve over here. So though it is rare, in pediatric oncology, it's a big problem because it's the #1 cause of cancer death in children less than 15 years of age. And this is again an age cutoff that I want you to remember because I will come back to it. So radiation works when we treat them for the first line with radiation. So it was logical to try to treat them again with radiation. And when we try to do that in SickKids first and then in other places, we did find that there was a survival advantage. It was prolonged to 7 months from the second progression, the first progression to the second progression. And the survival was increased to 23 months when we radiated these children again. And the 7-month number and the 23-month number is again important because this is the benchmark against which we evaluate the current generation of clinical trials that we are having for DIPG. So when we go into clinicaltrials.gov, there are actually 27 actively recruiting trials, and fortunately or unfortunately, majority of them are in phase 1, which means that we are just looking at safety, having some clinical or preclinical data to support that it's a flicker of hope or a flicker of signal. And I will be dividing these into 2 or 3 rather groups of interventions that are being tested. One is with inhibitors and molecules that are targeted against specific driver entities, some immune-directed approaches that are coming up, and some technological advances that are making us hopeful for the future. So coming to the first group of trials that mainly involve targeted inhibitors or drugs that actually metabolically reprogram the tumor and try to make them differentiate because it's a huge tumor in a critical part of the brain that you can't really take out. So not only do you need some cell kill, but you are hoping that there is some metabolic pathway that you can either activate or inhibit and allow those abnormal cells to differentiate and mature and become more like normal cells. So a list of unfortunately unsuccessful trials is what I need to begin with. As you can see, number of drugs mentioned here. I won't go into each one in detail, but just to make sure that when we design a clinical trial, we are aware of the things that are needed to be kept in mind. So when we use large molecules, they don't go into the brain. So even if you have an EGFR mutation, the drug may not go into the brain. That's why your survivals don't improve. Similarly, there are other targeted therapies that actually target a specific alterations which are not universal in this particular entity that we are dealing with. So we need to be mindful that the pathway that we are targeting is more universal, as well as the drug penetration issue that was alluded to is kept in mind when we choose a molecule to increase its chances of success. I will talk about ONC201, which was published in August 2023, so a couple of months back in Cancer Discovery, and there has been some controversy regarding it. It was an old molecule, so there were no commercial interests, and it was acting upon a metabolic pathway that allowed apoptosis to happen, and in parallel involved obliterating or inhibiting some of the pathways that were activated in the tumor. So taking you to the survival curves, it is how a clinical trial is designed that is very important. You can see that there is like hope when you see the survival curves, but when you go into the detail, you found that many of these patients were actually non-recurrent patients. The tumor was actually having some changes when you were treating them with the drug, which was preclinically established, but the survival after progression, after using the drug, was only 9 months, which was not very different from what we were having with radiation, which was between 7 to 9 months. Again, as I mentioned, the caveats of this particular trial, many patients were non-progressive. Majority were actually older patients, and there is data to suggest that older patients may have a different biology and they may have a slightly improved outcome. The location was again thalamic, and it is also known that thalamic tumors may have a better outcome than pontine tumors. And there was no actually mention in the trial that whether some patients got re-radiation, because then you have an effective modality and a second drug, so you don't know which is actually playing a dominant role. In the plots that show response, you can again see that the tumors which were recurrent actually shot up most of the times. There were some which responded. So the trial was criticized and had its like supporters and detractors, but it was something that the community was excited about and is still probably excited about to see how it plays out in the future. So we will have the trial open for older patients with thalamic tumors right across the street from SickKids in Sunnybrook, and we will be sending some of our older patients to the collaborators that we work with. So we will know in the future whether this drug actually plays out to its promise. There were other pathways that could be simultaneously activated in such tumors. And again, you can see some of the problems of sort of publishing data where re-radiation was given and there was a response thereafter. So you really don't know whether it's a drug that is effective or whether it's re-radiation that is adding to some of the benefit that was seen in these case series. This is, I mentioned, because this is a trial that is currently active and open in SickKids. It again targets a subset of patients who have the PTGFRA pathway activated or mutated within the spectrum of H3K mutated gliomas. So maybe not all patients will benefit, but a subset will. Again, it will be played out in the future whether it works in at least a significant bulk of patients. Some other promising preclinical data that is yet to be translated, and I just show this to see how the field is sort of changing. The HDAC inhibitors per se did not work, but now there is again regained interest in using molecules that may have a better CNS penetration and may have a more direct effect on the mechanisms that are actually defaulting in this tumor. Similarly, there was a recent paper published this year that actually used antisense oligonucleotides to actually allow the protein expression to happen. And it was promising in mice models, but again, it needs to be seen whether it can be translated in humans and cause benefits. So moving on to some exciting immune approaches that have come up in the recent or last couple of years. The GD2 expression in several DIPG tumors were found to be high. So one of the groups that is invested in this developed a CAR T cell, and it showed that there were some patients who benefited. Again, I have highlighted the age here. You can see there were 4 patients reported in this Nature paper, but this patient who responded was again an older patient. But again, the survival was after progression 10 months, so not much different from 7 to 9 months. And the total survival of 26 months, where we already with re-radiation had a survival of 24 months. So small gains, but lots more to do in the future. Similarly, the spinal tumor that responded in the same paper was in a 25-year-old, just to suggest that what is being sort of said as a successful trial, the term success is probably relative depending on which age you find these patients in, how they have been treated before, and what their presenting features are. But there were some things that we learned, that the immune inflammation was important. And if you could activate the immune system, there were some patients who responded, though DIPG has been traditionally thought to be a immune cold tumor when immunotherapy like checkpoint inhibitors really didn't work. There was a flicker of hope over here. Another group tried to target another molecule which was increasingly expressed in tumors, and they found some successes. Again, not magic numbers, but survival that was prolonged, including a patient that had actually histone wild type, and we know that their biology would probably be totally different. Thankfully, this paper at least acknowledged that the age of these patients was higher than what we usually expect for children with DIPG, and they specifically mentioned that no re-radiation was given, and they found some correlations with cytokine elevation in CSF with the responses that they reported in this trial. So the same group from Seattle is now targeting 4 antigens or epitopes by using CAR T. And this was initially exciting, but again, we need to see once this trial opens. It opened early in this year, and when it starts to accrue, whether it actually shows a flicker of hope. There were vaccines that were developed that, again, one odd patient responding after pseudo-progression. So each patient probably has some unique features that we need to go into deep, because each sort of molecule or each sort of approach has one of these survivors who do kind of well. But really, with the biomarkers not really well defined in some of the trials, we really don't know who benefits and who doesn't. So for the next generation of trials, that is something that we probably need to be mindful of. This particular trial found that CD4 T cell responses were important for response to the vaccine that was given. And another pediatric trial used a different set of a different approach to the vaccine, and found that the presence of T cells and lower myeloid suppressive cells were actually predictive of, again, a response in N of 1 patient that they reported very nicely over here, as you can see. So the last bit is to talk about technological advances. And again, here we have some of the things that we have here in SickKids. So this are basically based on focused ultrasound that are usually done by neurosurgical colleagues in SickKids and elsewhere, where they try to use a specific frequency of ultrasound to either disrupt the blood-brain barrier and allow older drugs that were not probably found to be useful, though they were successful in cell lines, they really didn't go into the brain. So there's a trial now open in SickKids and Sunnybrook using doxorubicin and focused ultrasound. There's a group in New York who are using the same focused ultrasound to give some molecules to the peripheral blood that they are using the ultrasound to cause activation in the tumor that can lead to apoptosis. This sort of approach can also be synergistic with radiation and can also have an effect on the immune microenvironment. So just starting right now, and we will know in the future whether these sort of approaches, which are more driven by our neurosurgical colleagues in association with our team, to see whether this is successful in the future. So just to conclude, this is from like a personal thing from an oncologist who is relatively young. So we see that there are several trials that develop or like speak of one odd patient who responded. So not only do we need to have robust preclinical data and early phase data to bring in molecules in the clinical domain, but we really need to incorporate biomarkers very carefully to see that whether we can identify who responds and who doesn't and why that happens. And I would even advocate that DIPGs, nobody was biopsying them initially. So we were like several years behind. Now we are biopsying and frontline, but maybe there is a case and the patient advocates need to be involved in that to have a biopsy when they fail the experimental therapy to actually understand why this escape happens. And again, seeing long-term, seeing the location of the tumor and the biology as far as we understand it, each of these one-off trials will probably have some successes, but ultimately we probably need to go the leukemia way, which is like the successful model for all of us, where we not only need cell kill, but also some sort of metabolic disruption, differentiation of the tumor because the tumor or the structure is not going to go anywhere, and harness the immune system to actually help that happen. So ultimately, we have to use these individual molecules which are promising and tie them up in a rational way, evidence-based way, to see if we can use mutually synergistic combinations to improve the real outcomes of the patients going from months to maybe years in the future. So I will stop here, and later on, I'm happy to take any questions. Thank you.
Superbly right on time. Well done. And so now we're going to hear about the genetics from a PhD student in Cynthia Hawkins' lab. Evan is going to tell us about what has been discovered in the last little bit more than a decade of a tumor that was never biopsied to a tumor that has been, and the genetics is really kind of super duper interesting. So over to you, Evan, and explain to us what's known about this.
Yes, thank you for having me. I will also try to be very on time, although I am a little bit sick, so please bear with me. And so today I'm going to be talking about some of the fundamental research on these tumors that have led us to the discoveries of some of the molecular drivers, which are currently being leveraged in the development of these very important therapies for these very deadly diseases. And so going back to the very beginning, our fundamental understanding of these molecular drivers begins with the understanding of histones. And so going back to our basic biology, histones are these nuclear proteins that are essential for condensing DNA into nucleosomes, and they regulate chromatin and transcription, and they consist of a family of proteins that interact together to create this core unit around which DNA gets wrapped. And so within the realm of DIPG, much of our knowledge of DIPG genetics revolves around one particular family of histone proteins that are the histone H3 families, consisting of their canonical histone H3.1 and its variant histone H3.3. And these mutations really, these histone proteins are really the basis of our understanding, like I said. And they represent key sites of what is considered the hallmark mutation of this disease. That mutation specifically, as has been introduced, is a substitution at position 27 on the histone H3 tail. It's a lysine to methionine substitution, and these mutations occur exclusively in the midline, most prominently occurring in the pons, and they are the site of interest. These histones are subject to a number of different modifications that can affect things like chromatin structure and transcription. And the prevalence of this mutation has led to the classification of DIPGs to also be known as the diffuse midline glioma K27M mutants, which have all been based on the discovery of these oncohistones, as we refer to them. Now, beyond this key mutation, like I said, these histone mutations occur in about 80% of these tumors, and they sort of revolutionized our understanding of the molecular drivers of this disease, although they are really the jumping off point. So each of these histone mutations, as was briefly mentioned before, is associated with a series of respective co-mutations that have allowed us to begin further dividing up these tumors into specific subgroups. And so amongst these already rare tumors, there are even more rare classifications. Those tumors possessing the histone H3.3 K27M mutation are often seen with recurrent mutations in very classic pathways such as P53, amplifications in things such as PDGFR alpha. And within the somewhat rarer group that possesses H3.1 K27M mutation, we see recurrent mutations in also pathways such as P53, but notably ACVR1, which is going to be a focus for the rest of this discussion here today. And so the discovery of these oncohistones and their co-mutations have allowed us to really begin identifying these well-defined subgroups of this disease, which is a very important for us to be able to learn how to not only understand but eventually treat, because each of these subgroups, as has been mentioned, has distinct differences between them in not only their anatomical location but also differences in their age of onset, their co-mutations, and ultimately their survival differences. And within the group, as was mentioned before, there's also a small subset that don't actually possess these histone mutations, although they share a number of similarities to their mutant counterparts, including not only the location, but some of the co-mutations that they possess as well, including things like ACVR1. And so this was really the basis of our understanding of the molecular drivers, and it has only been expanded every year as we progress in our in understanding the landscape of these diseases. And so one of the key mutations that we're going to be talking about today is the ACVR1 mutation. And these mutations are part of the secondary hits that occur after these histone mutations specifically. And they're a really unique case, and they have some qualities that can be leveraged, hopefully, for the development of therapeutics, one of which is that they're most often occurring, as I mentioned before, specifically in one subgroup, most prominently in the histone H3.1 K27M gliomas, but also in their location. As being part of that subgroup, we're really able to see these mutations arise in the pons itself. And so ACVR1 is a gene that encodes a receptor, kinase receptor called ALK2, which is part of a larger pathway. It's part of the bone morphogenic protein pathway. And it actually, ACVR1 mutations constitute the next most common site of alterations in these tumors. Histones are notoriously difficult to target, and so leveraging some of these recurrent co-mutations really provides a nice avenue for the development of therapeutics. And so this mutation occurs in approximately 30% of DMGs in total, but nearly 85% of these H3.1 K27M tumors. And it's a very interesting site of these alterations, where germline mutations in this gene often lead to a different disease known as FOP. which is a musculoskeletal disease. However, if the mutations are seen somatically, that's when we see patients present with DIPGs. And so, as was mentioned in the introduction, mutations in this gene are activating, and they promote a large degree of signaling through this BMP pathway. And so the upregulation of this pathway in these tumors is driven by this ACVR1 mutant expression, and it leads to further expression of other pathway members, such as ID1, ID2, and ID3, which are DNA binding protein inhibitors. And ultimately, what these do in the activation of this pathway aids in the acceleration of tumorigenesis while repressing cellular differentiation. However, as I said before, there is hope in the specificity of these mutations, just as emphasized here, in the fact that they occur so commonly with one specific subgroup of tumors and in a specific location in the brain. And so, understanding mutations in a particular gene is part of the story, but the other part of the story, especially in looking for therapeutics, is understanding the pathway that's ultimately affected by these mutations. And so in a canonical ACVR1 signaling, you get the formation of these heterotetramic receptor complexes that are formed between the different types of receptors in the BMP pathway. And upon the binding of their respective ligands, you get the phosphorylation of this glycine serine rich domain of ALK2, the encoded protein, which enables the kinase domain activation and subsequent phosphorylation and downstream signaling, which leads to a number of different processes, including cell growth, differentiation, regulation of apoptosis. But this pathway is also has the added complexity of these non-SMAD specific pathways being activated, including PI3K signaling and MAPK signaling as well. And so, although this is the canonical sense, like I said, there's a number of mutations that are present in these genes, and they occur at various sites within the ACVR1 gene. And over time, as we have furthered our understanding, we have noticed that there are a series of recurrent mutations within the DIPG pathology. So a number of these mutations occur specifically within the glycine serine domain or within the protein kinase domains, and these remain the hotspots for a series of substitutions. And although these mutations are diverse in their frequency, there are a few that have been notable in their prominence within DIPG, such as the R206H mutation in the glycine serine domain, as well as the G328V mutation in the protein kinase domain. And these represent the most common alterations, which ultimately will lead to aberrant increase in the pathway activation independent of ligand binding, which drives the expression, like I said, of downstream proteins and helps accelerates proliferation and tumorigenesis through ID1 and ID2, for example, while simultaneously repressing cellular differentiation. And what's really been interesting about the investigations into these specific co-mutations is actually seen quite nicely in the modeling of these tumors as well. And so with the ongoing development of these preclinical models, the role of ACVR1 in tumor initiation and in maintenance is constantly being investigated with models in multiple lineages, such as on the left here being driven in neuronal lineages, and on the right here being driven in oligodendroglial lines, that we're learning more and more about the contribution of this mutation as a major factor in the development of specifically these high-grade tumors, which ACVR1 has been shown to not only contribute to the increased incidence of these tumors, but also a lower overall survival. And notably, the cooperation between ACVR1 and the histone mutation and some of the other co-mutations that are also occurring. And so, DIPGs, although they possess a hallmark histone mutation, which allows us to categorize these tumors into distinct subgroups based on some of their incidence, their location, and pathology. Each of them is further characterized by these associated mutations, which represent distinct subgroups of the disease and allow us to classify them and hopefully find similarities that we're able to treat based on these classifications. And so among the histone mutations, H3.1 specifically, tumors possess a high incidence of co-occurring mutations in ACVR1. Like I said, it's nearly 85%. And these mutations occur predominantly in specific regions amongst within the gene, serving as a gain of function, leading to aberrant downstream signaling. And this signaling ultimately leads to increased proliferation. And so taken together, we're not only able to affect some of these prominent pathways that are promoting tumorigenesis, but to do so at the beginning of that pathway, which is really important. And preclinical models have also further emphasized this in the fact that these ACVR1 mutations seem to be really key in aiding the development of these high-grade tumors, and ultimately reinforcing that ALK2 is a key point of intervention for the development of novel therapeutics. And so I actually think I ended a little bit before time, which is nice. And I'll have happily be able to answer some questions at the end.
You in superstar status, young man. Look at that. Finished 2 minutes in advance. And you set the bar for Hector now. So Hector's going to now talk about specifically this protein kinase and efforts to try and develop an inhibitor of it, and one moreover that is known to get into the brain. And this is done in Rima's team, and it's upstairs for me, right up there. Okay, over to you, Hector.
Yeah. So hello everyone. Thanks for the invitation. So during my presentation, I'm gonna be talking about the latest effort that we have done here at OICR in order to develop ALK2 inhibitors. And these efforts are based on a conformationally constrained scaffold. So give me one sec. So before jumping into the actual part of the project, I just want to give a brief introduction about the history of some of the scaffolds that have been developed to target ALK2. So following the discovery of the dorsomorphine, which was one of the first molecules that was developed to target ALK2, different pyrazole 1,5-pyrimidine analogs were developed, and they proved to be useful in vivo inhibitors of ALK2. However, they also showed to be non-selective compounds, and they also presented some dose-limiting toxicity issues. So after these efforts, our colleagues from SGC, in collaboration with Harvard Medical School, they came up with the 3,5-diphenylpyridine scaffold, and they developed an extensive SAR that concluded in the discovery of this molecule, which presented suitable brain penetration and also suitable pharmacokinetic properties, low cytotoxicity. However, it also showed to have a low potency of 115 nanomolar, and well, that kind of sounds like pretty good, but for kinase drug discovery, that's kind of high. And also, it has some selectivity issues for ALK2 against ALK2 over ALK5, with just a 70-fold selectivity. And well, I just want to remark the fact that we want to try to prevent inhibiting ALK5 because the literature reports indicate that inhibiting ALK5 can cause serious gastrointestinal problems. So after the discovery of this molecule, the OICR drug discovery team, in collaboration with SGC and M4K Pharma, became interested in developing an additional SAR expansion of this molecule. And after a lot of big efforts, David Smil and co-workers here at OICR realized that by changing this methyl group from the second position of the pyridine to the fourth position of the pyridine, they could achieve to generate M4K 2009, which was not only a more potent compound than the previous one, also showed a higher selectivity for for ALK2 over ALK5. So this was really exciting, but as Alex said, we always want to generate more potent compounds and more selective compounds. So then after this effort was done, a former master student from our lab, Diva Ensan and David Smil, analyzed the way that the ligand was binding into ALK2 based on this co-crystal structure. And they rationalized that by joining these two carbons with different ring systems, they could actually freeze the bioactive conformation of the ligand, and maybe they could generate some more potent and selective compounds. And well, actually some recent medicinal chemistry reports have shown that the rigidification of a ligand can actually lead for an improvement on potency and also an improvement on selectivity. And also, mostly important, it can also increase the permeability of the compounds. And this is really important, especially for this project, because we have to keep in mind that we are trying to develop CNS drugs that actually have to be able to permeate the blood-brain barrier. And that's one of the biggest challenges of this drug discovery field. So actually, maybe by just joining these two carbons with different ring systems, we could achieve to have that. So that was the hypothesis of the project. And that was actually when I joined the drug discovery department for my PhD. So Diva and I synthesized a small set of compounds based on M4K 2009. As you can see, we introduced a 5-member ring, a 6-member ring, and different 7 member ring systems with different heteroatoms in between the linker. And we also tried to vary this position of the molecules. And well, one of the biggest challenge of this project was to be able to synthesize these compounds in the proper amounts, because each of them required a specific synthesis path. And at the very beginning of the project, we thought that we could generate these compounds in a decent time, but it took us longer than expected. But at the very end, it was worth it. So after we finished the synthesis of the compounds, we submit them for the biochemical kinase assay at RBC, to the nanobread cellular assay with our colleagues from SGC Oxford. And we also performed some in vitro ADME here internally at OICR, made by Dr. Taira Kiyota and Dr. Ahmed Aman. And well, normally when I give this talk, I go through the whole data set, but because of the constraints of time, I just summarize the most important information for the most promising compounds, which are these 4 compounds, these ether-linked analogs with the oxygen in the down part of the linker, and these N-methyl analogs in the middle part. So as you can see here from the biochemical kinase assay, from this green part of the table, these 4 analogs present a higher biochemical potency when compared with 009. In terms of selectivity, in biochemical selectivity, the ether-linked analogs had a very similar selectivity fold when compared with 009. But if we compare it against the N-methyl analogs, we can see that they are way more selective, actually with a 5 selectivity fold increase. And we also saw this trend in the cellular assay. We also saw that the 4 compounds were more potent than the parent compound. And also we saw an increase on the selectivity on the cellular assay. So we were really happy to see that. And then we performed the mouse liver microsome and human liver microsome to see the stability of the compounds. We saw that 3 out of the 4 compounds presented medium to high stability. And for the Keiko-2 permeability assay, we saw that the ether-linked analogs showed good permeability numbers, while the N-methyl analogs showed a decrease on permeability. And we attribute that to the fact that now we have 2 tertiary amides which can be protonated, and that could be affecting the permeability. So after we obtained all this data set, we proceeded to investigate the in vivo exposure of our new set of compounds and compare it to M4K 2009. So in this graph, you can see the results from this assay, where you can see the concentration of the compounds in plasma for each data set point where blood was extracted from the mice. And as you can see from these results, unfortunately, none of our compounds presented higher exposure than the parent compound 009. However, we saw that this compound 2281, which is the blue bars, presented a similar, a little bit higher exposure than 009 in the first 2 time points. However, then it has a quick decrease. And well, this was maybe not that surprising for us, but at the same time, we were not demotivated because there's still more work to be done in this part of the molecule where we can try to fix the pharmacokinetic properties and increase the exposure of the compounds. And we also decided to go and test 2 of our compounds in BBB penetration assay, where we calculated the brain to plasma ratios. And because, well, this is really important. Actually, this is because it's one of the main challenges for these CNS drugs. And as you can see, when we compare our new 2 compounds against 009, we see a brain to plasma ratio, higher brain to plasma ratios than the parent compound. And well, this is really exciting because that means that actually by the rigidification of the compounds could actually increase the permeability, the BBB penetration, and specifically for these N-methylated analogs, which we were not expecting because we saw the exposure wasn't really high, but they at least in this assay they showed to be good brain penetrants. And well, just to conclude my presentation, I just want to show this beautiful image, which is a crystal structure of the N-methylated analog in the ALK2 pocket. This was obtained by our colleagues from SGC, and the co-crystal structure was analyzed by Dr. Laurent Hofer here at OICR. And as you can see here, this is a clear image that reflects that the hypothesis was actually accurate. We were able to preserve the way that the M4K 2009 was binding to the ALK2 pocket. So the M4K 2009 is the green compound here, and overlapping is the yellow compound is our N-methylated analog. And as you can see, both of the compounds are overlapping themselves, and they are actually making the same interactions with the residues of the ALK2 pocket, and which actually shows that we were able to freeze the bioactive conformation by putting this 7-member ring system. So in conclusion, the rigidification of the ligands using a 7-member ring system actually helped us to induce a conformational lock of the bioactive conformation of the compounds. This led us to have an improvement on the potency and selectivity for ALK2 over ALK5. And we were really excited to announce that these 2 compounds are the most potent ALK2 inhibitors reported to date in the literature. And we still have room for improvement. We can still modify this moiety from the compounds to increase the pharmacokinetic proteins of the pharmacokinetic properties of the compounds. And something also that is really exciting is that these SickKids scaffolds are unique amongst type 1 kinase inhibitors, and they also open a novel chemical space for further optimization and also for future evaluation in orthotopic models of DIPG. So this opens a kind of like a new chemical scaffold to develop new ALK2 compounds. And yeah, that's it from my side.